No, it's the first trick is to have a very good OCR program.
Oh, yeah.
Anyway. I wouldn't want to do Chinese though.
No. Yeah.
At least with French and German, the letters are pretty much the same, so I can spot spelling errors that the OCR makes.
Oh, yeah.
Okay, I'm going to be able to do Chinese. Steve McGrew is going to try to join us today. We'll see if he actually succeeds.
Okay. Sounds good. I haven't seen him in a while.
Yeah, maybe we get him to do a presentation.
Yeah.
Okay. Like what were the FPGAs? He's good at programming FPGAs for machine learning.
Oh yeah.
Yeah.
Okay. Maybe we can get him to do it.
I think I just heard something about FPGAs, I guess.
Oh yeah?
Yeah, that's what I was working on with my robotics project for the motors thing. So we wanted to have a really fast interface, so we could do most of the computation on outside the robot and not on the robot. So we shifted to FPGAs lately, and it has been a challenge to get it work initially, but yeah, the performance have been fantastic. So that is what I have been doing for the last one and a half.
Oh, okay. But Steve's probably a pioneer in this. He's retired from running. He's a physicist. He's retired from running a business, a holography business, which included selling machine learning software and FPGA stuff.
Okay, hello everyone.
Hi sir, how are you?
Right.
How's everyone?
Okay. Well, welcome to today's meeting. Welcome everyone. I see we have a bunch of people coming in at once, so I don't know. We might get a few more people come in, and so I don't want to necessarily hold things up, but they might be coming in. First, I wanted to ask: does anyone have any news that they have? Any events going on? Let's see, look in the chat. Oh, we just had people say hello there. Okay, anyone? No one has anything to share? I have a couple of things to present. We don't really have any presentations today or anything, but we can talk about. Well, let me start off with a follow-up from last meeting. So I ended up putting together a couple of documents here. Let's see if I can present now. And this was based on our conversation of what kinds of tasks we might want to work towards this year. So if you can see my screen, it's coming up here. Okay, I'm presenting. Okay, so this is it's just a beginning take at this, but this is a project board in GitHub for some of the things that we've mentioned in the last meeting. So I think this is the right one. It might not be the right one, actually. Let me check. I think it's in this one. I made that one, then I closed it out. So it wasn't saving the tasks properly. So this is it. So if you go to group meetings, that repository under projects, you'll see there's major tasks for 2020, and I'll put this link in the chat. Okay. And this is basically the task board for, well, we'll just keep this up when we mention something in the meeting. We'll put this on as a task. And then, there are 4 columns: to do, hold, in progress, and finished. So if we someone starts something and it's in progress, we'll put it in this column. So we talked a little bit about the CompuCell 3D model last time. I put it in progress because I know that Ujjwal has he's been working on it a little bit. And then the rest of them would be to do. So whenever you generate an issue, or we'll mention it in the meeting, or maybe we can talk about it offline, and I'll put it in this table so that everyone can see it, and then we'll track it. So I have a couple here. Embryo visualization for Docker, we mentioned that. So, we can update accordingly. Axolotl montaging, that's something that's to do, but we don't really have any progress on that yet. The Basilaria non-neuronal cognition paper, this is the one we talked about putting an outline together for. And I haven't been able to get that outline together yet, or we haven't been able to make any real progress on that yet, but it's it's coming. And then this complexity measures, which is something I got out of old file. So I'm kind of also in the progress of going through some of the things that we've talked about in previous meetings. Maybe this is like even before DevoWorm ML, and just kind of bringing them back up into this queue so that we can kind of bring them to the front of our mind. Steve McGrew via yahoo.com. Oh, Steve can't connect to the Hangout or the meeting. Does he have the right link?
Yeah, I'm getting the tiny URL.
Okay. Huh. I don't know. Sometimes you have to try a couple times because sometimes it won't let you in for some reason. And you also have to have a Google account. So
You have to have a Google account. Who's calling by Yahoo? Maybe that's the problem.
Well, I mean, you have to have a Google account, so it's just something.
So that means you have to have a gmail.com email account?
Yeah, you do. I think he probably does. I mean, most people do, but yeah.
Okay, I will reply to it and I'll CC you.
Okay.
Okay.
So these are the major tasks, and so we'll just go through this once a week, at the beginning of the meeting and see where we are on things. So scheduling presentations, I put that on hold because I have a couple of people in mind. I've yet to do that. Actually, that's a good, Dick mentioned at the beginning of the meeting that we should also get Steve McGrew to give a presentation. So Steve McGrew, he was an early collaborator in the group, and he's given a couple of talks in the past, but he's been busy in the last couple of years, so he hasn't been in our meetings. But I mean, I think he'd be interested in rejoining periodically, at least, and definitely maybe giving a talk on FPGAs, which are a type of hardware for machine learning. That might be him. There he is.
Oh, you got in?
Yep, you got in.
Oh, good.
Hello, Steve. Oh, you're muted. Oh, he left. I think he's gonna come back in a minute. It's a typical sort of glitch in these kind of meeting spaces, but let's see. So just to finish up with the major tasks, so that's where we're going to keep things. And if you, I think if you want to join, if you want access to this board, I can add you into the repo as a collaborator. So let me know if you want to have access to this board. I can put you have to have a GitHub account to do it, but I can add your GitHub ID and you can update the board yourself. Otherwise, we can do it at the meetings. So the other two things I have are the weekly agenda. So this is a list of the things that we have going on. So, on a weekly basis. Oh, there's Steve McGrew.
Oh,
Back in. So this is the weekly agenda, and this is starting this January. So again, like I said before, we have a bunch of spots open. I've kind of broken this out by week. And for previous weeks, of course, I have the recording here. I have the topic. And then if we have someone, we can schedule someone. I have myself down for a couple things, which will be, those are kind of to be determined, but I just want to put them on the schedule. So this is for like your reference, for if you want to know when to schedule something or if you want to find something from a previous week. So that's at that link, and that's shared with everyone in the group or just in the general public domain. Also, the list of potential papers, which is another GDoc file. This one is so I just have a couple of things on this list right now, but there'll probably be more things. This is basically papers and blog posts that we've talked about. So this is the bacillary and non-neuronal cognition paper. We need an outline for that. Meta-organisms, which is something that I'm kind of kicking around with another collaborator, but we might talk about in a future meeting. That's somewhat of an outline, but we'll talk about that more. And then transfer learning, which is something that Vinay proposed, a blog post. We're still sort of on hold with that. So, we'll have if there's an outline to review what the status is, if it's a blog post, because then we can sort of figure out, maybe we'll just move faster on that and put it out as a blog post. We have a preprint if we're going to do that, or the ultimate venue, which is like a book chapter, paper, like a peer review publication or whatever. So we have all these things being taken care of here. I could talk, let's see. Well, there's Ujjwal. Hi, Ujjwal. You don't have your face online too much here. Okay. Dick Gordon says, I could talk on Gordon 2020, the whimsical history of the proposed motors for diatom motility. That would be pretty good if you could give that talk. When do you want to do that? What week? Okay, well, yeah, we could probably do it next week. Jesse, but I don't know. He hasn't been feeling well this week, so he might not be able to make that one. Maybe I'll put you down for next week, and then we'll see if I can edit. I can't edit it right now, but I'll put you down for February 17th when I can edit this. I don't know why it's not letting me edit it, but and then move maybe Jesse the following week, and then I'll present maybe the next week. We'll figure this out, but plan on February 17th on this week.
okay.
And then we can, and if you have anything, like any readings you want people to do beforehand, send them out to me and I'll send them out to the group by about Thursday or Friday. I mean, I can send it out later, but, just give people a chance to read it. So, welcome, Steve. I haven't seen you in a while. We haven't seen you in a while. I wanted to make a offer here. We're talking about giving presentations to the group. Would you be interested in giving a presentation to the group? Okay. Dick says, yes, we could just use critical readers. Just finished writing last night. Okay. So, Steve, I asked the rest of the group if they would like to present something, and I extend an offer to you to present on maybe something that you're involved with. Dick suggested your work on FPGAs, which might be interesting to a lot of what we're doing in the group right now. But it could be on any topic you want. So I don't think you don't have to answer right now. You can think it over. We can talk about it by email. And just to let you know, just to extend the invitation. And Vinay said that he'd be interested in presenting something as well, or is this in response to Dick's work? I wasn't sure what he was responding to. Okay, so those are the three things that we have that we put together for keeping track of projects and things like that. So we'll revisit this weekly and see what the progress is on things. It's just good sometimes to go over them, again and again. It keeps people on track because they keep thinking about it. So I guess today I wanted to talk a little bit about some things that I've found online, some things people have sent me, papers and videos and things like that. Dick says, okay, I'll clean up the figures now out of order and send Bradley a PDF for distribution. Okay, yeah, I look forward to that. So let's see. First thing I wanted to bring up was something interesting I found in my reading. Or this is a tweet on something that. So I don't know. Most of you probably don't know this. Maybe you do. Roger Sperry was a neuroscientist. He was active in the 1940s, 1950s. And when he was, one of the things he's known for is doing this experiment where he, okay, Vinay says, it is just a blog post, but I'm able to make less progress on it. As I got a bit busy with college and stuff. Final year isn't as easy as I thought. Well, okay, well, that's good. We'll go keep revisiting it and see if you can do it. Fine, well, but yeah, definitely. I'm glad that you're doing well in school. So, okay, so this is the frog. Let me show you this experiment. So this was an experiment. This frog. Let's see if I can get it to run here. Okay, I'll just run it like this. Alright. Okay, see this frog is picking off this this fly. And this is where the frog is not able to locate the fly. So there's a fly, and the frog kind of does the side of the fly if you introduce the fly direction, whereas before it was picking off the fly with pretty good accuracy. Now it's almost like it's avoiding the fly. So why is it doing that? And like I said, Roger Sperry was a neuroscientist who did this experiment in the 40s where he took the eye of a frog and he rotated it 180 degrees. So the idea was that you have the eye and you have something that the eye is mapping to in the brain called the tectum. And if you take the eye and you rotate it at 180 degrees, it rewires the tectum and it changes the way in which the brain is wired, and it changes the visual reference frame of the frog. There's Jesse. Okay. Got another person in, Jesse. So I guess, yeah, if you want to turn your camera on, I wouldn't do it all at once, but just so Steve can see you. Steve is, this is his first meeting of the year, so he hasn't met a lot of you. So if you want to chat and say hi to him too, go ahead. Okay, so this, this frog, the eye was rotated 180 degrees, so the brain was rewired in such a way so that it changed the visual reference frame of the frog. So when a frog goes and tries to get a prey item, like a fly, it shoots its tongue out, and it's usually able to hit the fly with pretty good accuracy. It's pretty well adapted to do that. But when you shift the reference frame, and you shift the eye in this case, it actually changes the reference frame, so it no longer can make that connection between the visual input and the motor control. And so they talk about chemoaffinity in this. What that means is that there's actually something that goes on when the brain is being rewired. And this is the best diagram I could find of this that explains the whole thing in one shot. So this is a picture of the eyes here, and this is the tectum. So you have the eyes, and they cross hemispheres so that this eye is, projects to this part of the tectum. This eye projects to this part of the tectum, and this is how the visual representation is built in the brain. So this is integrated. This visual information is integrated eventually with motor information, and the frog can flick its tongue and catch the fly pretty quickly. This is the original state here at the bottom. So there's a cross hemisphere. It's crossing the opposite side of the brain. And that's the way the visual representation is usually represented. So then when you do this surgery, you take the eye, you cut the axons here at the back of the eye, and you rotate the eye 180 degrees. Now, these axons have to remap themselves to this tectum, so they have to project back out. But how do the axons do this? And it does it through something called chemoaffinity. So the axons are then moved into a different position or a different orientation. They have to find their way to some sort of new mapping to this part of the brain. And so they use chemical signals to do this. So, the way the brain is set up, it's actually doing this in a way that actually changes the reference frame. So it's actually projecting in at a different angle. It's maintaining its connections, but the connections have been shifted somehow. And so it's building a new map of space in the brain. And now when you have this frog goes back out and tries to catch flies, its tongue goes in the opposite direction. So now you can see in this diagram, this tongue shoots out in this direction instead of towards the fly, even though the intention is to catch the fly. It always has been. But now the visual frame is rotated, and the tongue is going in the opposite direction. I thought I'd bring that up because, we've been talking a little bit about that in my other group. Plus, I thought it was a really cool experiment, and it has some relevance to development. Because, it does show, like, you could rewire the brain in development, and there's a lot of developmental plasticity that's involved in this. So I just thought that was a nice little experiment to highlight here. And then there was this response to the first tweet that I showed you, something to the effect of a similar turning away effect is seen when the ocular motor muscles on one side of one eyeball are cut at birth, preventing the usual experience-dependent programming of tectum maps. So in this case, if you cut the connections of one eyeball at birth in the frog, it can prevent this programming sort of in development. So that means that the tectum maps are modified in development and not allowed to develop as they normally would. Frog tectum is especially interesting in that it doesn't program saccades, which are rapid eye movements that organisms make. Usually it's involved in tracking prey, but it's also, you also move your eyes sometimes when you want to look at something else in the environment. So that's what a saccade is. But it does use that same build to a decision threshold population coding, which is a form of like how information is represented in the brain, to trigger the fly snagging action of its tongue. Snake fang strikes operate in the same way. So what they're saying here is that there's a sort of a representation in the brain that's used to sort of activate this fly snagging action. So even when you change how the information is mapped into the brain, we still have this same mechanism working, and it's working just in a different way because the map is different. So, and maybe that was a little bit above people's heads, but it's just an interesting experiment I wanted to talk about, and there are papers that we can talk about later that might clarify that if people are interested in discussing that topic further. But I just thought it was neat to show. So I also want to talk about some papers that have been been building up in the reading queue here. Let's see. So the first one we can talk about is organelle size scaling over embryonic development. And so I thought this was interesting because we did a paper a couple years ago on sort of the size and the distribution of cells in the embryo. And we built trees out of it. And it was a matter of like trying to figure out the size of cells and how they, their size upon division. And so it turns out that this is a literature in and of itself, and they call it size scaling in development, or in embryonic development. And in this paper, they explore this idea of organelle size scaling. So the idea here is that you have like a single cell, and it divides. And sometimes, generally speaking, the cell divides into two cells, and the two cells are then half the size of the original cell. But that's not exactly the way it always works in development. You get growth of the embryo, and you get growth in these cells in terms of their size. So you have a very small embryo, but you end up with a larger larva, or a larger, something going on in the egg that's larger than the single cell, and then eventually you have a juvenile that's much larger than the original cell. So how does that all get regulated? And what they argue here is that there are all sorts of scaling mechanisms that work in the embryo and in development that control a lot of this. So in this paper, this is a review paper, and I'll send out a link to this later in another email. But they basically review the literature on this, and they review a bunch of model organisms. So they review data from C. elegans, Drosophila, or fruit flies, Xenopus laevis frogs, and Mus musculus, which are mice. And they've provided insights into the developmental size scaling of the nucleus, mitotic spindle, and chromosomes. So all these structures apparently scale in such a way that allow for growth of the cell, or growth of the cell size, growth of the embryo, and so forth. And so they do, they do a bunch of stuff with data analysis in this review. So it was kind of weird when I read it. I thought it was is this a review or is this a data analysis? And it turns out they kind of do both in this paper. They talk about a number of different things. So basically we're talking about size regulation and how this works. So there's a bunch of information in here about scaling in terms of cell size, in terms of organelle size, and then they compare it across organisms. Let me go down to some of these graphs. So these are some of the images that they have. I don't know if this figure makes any sense on its own, but So they look at nuclear scaling, they look at spindle scaling. So there are different things going on inside the cell that they can track over time. And I kind of wanted to get, okay, so they kind of propose a model here. So they have models for developmental organelle size scaling. So here they have early development and later development. And they have limiting components and an equilibrium balance. So they're saying that there are changes that go on in terms of scaling that sort of regulate this process. So we have limiting components in the cell, and if you increase or decrease the size of certain organelles, it results in other trade-offs later in development, and that's how you get not only changes in size, but you get diversity across species. And so, I mean, I can leave this up to people to read, but I just want to preview this paper. I think it's fairly new. Let's see what there's another one in here. I think I wanted to get to a graph to show what scaling looks like. So this is kind of the idea here. So this is a graph of developmental stage. So you have your one cell, your two cells, you go up to you do cell doubling, which is exponential, and this graph goes up to 4,096 cells, and then you have different stages of development, which are just they try to normalize this over all of the different species. So they have worms, yeast, which doesn't really do anything. It's a kind of a control, and then they have mice, sea urchin, starfish, zebrafish, frogs. So they have this N over C volume ratio, which is a nuclear cytoplasmic volume ratio. And they look at that as their sort of variable, their independent variable here. And so they find that it generally increases during early development in different species. So you can see with mice, this volume goes way up after the 16 cell. In frogs, it remains pretty stable, but it goes up in later development. In starfish, it kind of increases, in between frogs and mice. And sea urchins are a little bit faster than starfish. But they use this volume ratio, which is scaling two features in the cell against one another. And that's generally how people do these scaling studies. They look at sometimes they look at things like brain size and body size. That's a very common scaling. And they get a very systematic result. They get some sort of function that they can then describe. Why is mice data cut off at 16 cells? I'm not really sure. I think that it might not really fit into their scheme here. It might go off the graph because it seems to increase quite greatly here. But I think they were showing it as like a comparison, like in this part of development, for different species, like how do they compare? So most of these are not going off the graph, but mice do. And worms, I guess, are increased, but they don't go off the graph. Well, maybe they do up here, but the idea is that you have these ratios that you can look at, and they have the systematic relationship between species usually. It's hard to really find a wide variety of species, but you do have this pattern across different developmental stages. So the data for figure 5. I hope this thing doesn't crash on me. All right, there we go. All right. I'm having a little bit of trouble here with I have too many things open, I think. Okay. My file might have, my PDF program might have crashed. Oh, there we go. Well, let me close down some of this stuff. So are there any observations on that, or? close some of these things here. Having some trouble with the memory on this. Found first paper. Not sure how much farther I'm going to be able to go. This way. I think my computer is needs more memory actually, but so Jesse, we have a bunch of chat items here. Let's see. Okay, good. Okay, let's see. The last message was from Ujjwal, and I just got that one. So let's see. Let me stop presenting. Okay, so Dick Gordon, 9:27 a.m. Human egg starts with 70 micrometer diameter. Okay. Why is mice data cut off at 16 cells? And then we answered that one. And then Dick also said there may be a change in implantation which differs greatly between mammal species. That's probably true. I think I'm frozen out here.
X-rays that went along that path, you know, that certain number of photons got crossed.
Yeah.
So what you do is each computer that's dealing with one ray would simply take the current values of all the pixels in that along that line and check their sum against what the data is.
Mm-hmm.
And if the data is higher than the sum, you add some. If it's lower, you subtract some, or you can do it in proportions. Okay? So then you'd have all of these little computers, each one dealing with its own ray on the same picture. Okay? This might converge very rapidly. It would be stochastic in nature to some extent. But it would be quite powerful, and it would be sort of simultaneously asynchronous parallel computing on a massive scale.
That would be fun.
Okay? And I haven't seen much on asynchronous parallel computing since maybe it's from lack of looking.
It would almost have to be from lack of looking, because it would be hard to be synchronous in doing your computation on the internet, in the cloud.
Oh, yeah, I suppose. That would be one way. Yeah, I hadn't thought about that. Yeah. Okay, so you're saying sort of crowdsourced computation.
Yeah.
Basically be asynchronous.
Right. Yeah, and there are utilities available where you can, there's clouds of computers you can harness to attack given problems, as long as you know how, which I don't.
Okay, any of you guys listening know Bradley's back. Know how to use the crowd for parallel asynchronous computing?
I see Bradley's muted.
Okay.
Okay, there we go. Yeah, it just muted me when I came in for some reason, but.
Okay.
So what are you guys, yeah.
We've been, in your absence, we've been discussing, parallel asynchronous computing.
Oh, yeah.
And Steve said that that's kind of the model for computing, using crowdsourcing.
Okay.
Because on the internet, you're not going to get any synchronicity. Okay?
Yeah.
So we discussed a particular model, which is to do computer tomography, where each computer is dealing with one ray across the image, and just trying to keep the sum of the pixels constant, while all the others are changing. And the idea is that this might converge very rapidly and allow a massive computation with processes.
Yeah.
Okay?
Yeah.
So, so I was trying to ask the guys listening if anyone has any experience in dealing with how to program a massively parallel problem that's crowdsourced. You still there, Bradley?
Oh, yeah, I'm here. I was just wondering if people had had something to add to that.
Yeah.
I know in terms of myself, I'm not really sure. I've not done a lot. Oh,
Okay.
so Vinay wants you to type it out. Yeah, I've not really heard of a lot of. I'm familiar with crowdsourced type sorts of research where you're getting people from all over to sort of solve a problem. But I don't know. Yeah, I don't know how. I couldn't really give you a good answer on that. Yeah, I don't know if anyone else here has any experience with that.
I'll do some searching and see if I can come up with some doors to peek through.
Yeah, definitely. It sounds interesting. If I guess first of all, there's a model of computation, and then there's like the application. So I don't know what even if it's a different application, it's probably would be an interesting thing to follow up on.
Yeah, apparently it's called cloud computing.
Oh yeah, cloud computing. Not really familiar with that literature too much, though.
And basically, it's a way to harness lots and lots of computers or spare power at data centers, that sort of thing.
Yeah.
You can just rent time on it. So rent a few minutes on a mega supercomputer.
Yeah. They do with they have like Amazon AWS and things like that where you can rent space, and then if you're at a university, you can do like use something like Blue Waters, which is what they have. It's run by University of Illinois, but they let institutions rent time on it. But you can do it remotely. You don't have to go to the computer center to use it. And then, but I think there's a lot of like stuff in terms of like a lot of innovation in cloud computing. People have kind of worked out a number of different models, but I'm not familiar with those. It might be something to review at one of the meetings. Oh yeah, Vinay actually maybe knows something about cloud computing. I don't know, Devansh, do you know anything about cloud computing? Or Ujjwal says, we do use servers for a big computation, but don't know whether it falls in that category. Yeah, there's probably a bunch of different terms they're using into the massive category. Yeah.
Same as Ujjwal, I do use like a lot of virtual machines on cloud for computation, but the distributed part of it is slightly different. It has components like OpenStack, which merges many different computers together into like a simulated system. So that is a little more complicated. That's not something I know much about. But for using single machines for computation in multi-threading over them, that's something we do all the time.
Okay, how about adding a project computed tomography via cloud computing? Yeah, we could do that. I'll put that on the list. Not sure I can get onto the... Yeah, I closed down my tab, so I'll put it on after the meeting, though. Then we'll review it next week, see what the status is. Yeah. So let's see. Next thing I wanted to get to here, we're about 10 minutes away from the top of the hour. So Jesse, I know you've not been feeling well, but I did have that paper that you sent me. I thought it was interesting. The direct fit to nature paper. And let's see. I don't know if I should share my screen again because this thing is unstable. But you don't have anything. I know. Yeah, I probably don't. But I just wanted to bring this to people's attention. And we could probably do something like later on. Oh, I guess you weren't here at the time, but we were talking about, scheduling time for talks. And I didn't know if you would still wanted to do do a talk offline sometime, maybe like sometime a different time in the week, if that worked better for you. Okay. So maybe we'll plan within the next 2 weeks. I can follow up on it. Okay. Actually, if you don't mind, I'll just go over the paper quickly and kind of share a little bit about it. If I can share my screen again here. Because I thought it was interesting. And then maybe you can give a longer presentation. That's kind of why I do these little mini reviews, so that we can get some ideas flowing. And anyone else is welcome to do this too. If there's a paper you read that was really interesting, you can do like a 2 or 3 minute presentation, just to kind of go over it a little bit with people. So this is a paper that Jesse sent me, and I actually found independently of him. Uri Hassan is a neuroscientist, and he's done a lot of stuff on neuroimaging, but this is a evolutionary perspective on biological and artificial neural networks. So the argument of this paper is that evolution is a blind fitting process by which organisms over generations adapt to the niches of an ever-changing environment. Does the mammalian brain use similar brute force fitting processes to learn how to perceive and act upon the world? And then he talks about deep neural networks, and they've exposed the power of optimizing millions of synaptic weights to map millions of observations to objective functions. And then these models do not learn an explicit human interpretable representation of the underlying structure, meaning they don't have it like a model in their network of the data. Rather, they use local computations to interpolate over task relevant manifolds, which means basically that they just find patterns and they lock onto those patterns in the data. Furthermore, counterintuitively, over-parameterized models, similar to evolutionary processes, can be simple and parsimonious. So he's saying that you can use over-parameterized models as a way to sort of reach robust solutions to a diverse set of problems. In contrast to traditional scientific models, where the ultimate goal is interpretability, over-parameterized models eschew interpretability in favor of solving real-life problems or tasks. So again, these models are not general, like we think of scientific theories, but they're very specifically trained for specific things. So like in machine learning, often you would build you might train a model to analyze a certain type of data, like, image data for cars or something, which is kind of what they're getting at here. We contend that over-parameterized blind fitting presents a radical challenge to many of the underlying assumptions and practices in computational neuroscience. So he's talking about computational neuroscience here. And it's an interesting paper. He talks about sort of how to sort of conceive of models that, that we use for analysis of data and maybe tying that to the brain. So this is kind of the idea here. If you have a model that underfits the data, you can use two parameters. You can get an underfit model. It doesn't describe all the data, but it describes some general trends in it. An ideal fit would be to use 3 parameters or something like something with, like this type of an equation. And then, you can move up to 20 parameters, which is an overfit model. In other words, you're describing too much of the data to be used in a general sense. So this is a 20 parameter model where you're basically, fitting a function to describe every point or almost every point in the model. What he's saying is that you have this new class of model which he's proposing, which is this direct fit model, which is where you're using 1,000,000 parameters to describe each position or each data point. And you can use these type of models to describe very specific types of data instead of using a generalized description. So if you have a very small amount of data, you can use this ideal fit extrapolation method. But if you have a lot of data, you can use this direct fit interpolation method. So you need a lot of data for this model, this type of model. You need a lot of parameters, and you can actually interpolate a direct fit to the sort of the natural structure of it. But you need a lot of data to do that. And then he shows in this where, if you have a distribution of data, that it only works in a certain portion of that distribution. This is what he calls the interpolation zone. This is the extrapolation zone. And that corresponds to the number of data points you have in that region. So he's arguing that direct fit models respond to things that we know, like artificial neural networks and BNNs, which are, I don't know what BNNs are, but biological neural networks. They rely on dense sampling to generalize using simple interpolation. Dense exhaustive sampling of real-life events, which the field colloquially refers to as big data, effectively expands the interpolation zone so as to mimic idealized extrapolation. And so a direct fit model will generalize well to novel examples in the interpolation zone, but not generalize well in the extrapolation zone. So it actually does generalize well in a certain region of space where you have a lot of data. But in general, these direct fit models don't generalize. And so this is a very, I think we need Jesse to present in more detail on this. And I think he definitely will make more sense when he does his presentation. But it's definitely a good paper to read over and, maybe try to make sense of yourself, because I think it has a lot of interesting stuff in it. So there are a lot. I'm not going to get too deeply into the figures, but that's basically the idea. So Jesse is wanting to present that in the next couple of weeks, and I'm going to schedule a time. He's going to present offline. So let's see, there were a couple of comments on this. So Jesse says, sure, he'll present in the next 2 weeks or so. We'll find a time offline, and I'll present the recording in the session because it doesn't work well for him to prepare and do something for this time. So it's fine. And then Steve says, this relates directly to structuredness. So yeah, Steve, we've been talking, you haven't been in the meetings recently, but we've been really on a kick of like talking about machine learning and structured data and unstructured data. And so that's definitely something, if you have some thoughts about that, about, how to apply technology to that, or if you have any things in your past that I know you worked on genetic algorithms, definitely that's something to share with the rest of the group. We'd be interested in hearing about that.
I'd be willing to put something together for that.
Okay, yeah, that's good. Well, we can talk about that more later. Maybe we'll put you on the schedule. But yeah, I look forward to it. Thank you. So anyone else I know this was a little bit, it wasn't like one theme in the meeting, so I hope people were, a little bit enthusiastic, they found something that was enthusiastic to be enthusiastic about. Okay, Dick, thanks for attending. And so we're at the top of the hour, so thanks for attending, everyone. If you have anything to add, I'll be available offline, and talk to you next week.
Thank you. Bye.
Thanks. Bye.